Depression is a common, debilitating and sometimes fatal mood disorder which will affect almost 1 in 5 of us over the course of our lives. The World Health Organization has stated that depression will be the leading cause of disability worldwide within the next decade.
Although there are many drugs available to treat depression, almost all target the same system within the brain, aiming to restore the balance of a set of neurotransmitters called monoamines. However, these existing medications have several problems. It can take up to 4 weeks for them to have any effect, and they are ineffective in a large number of patients.
So there's a clear need to develop new, quicker acting and more effective antidepressant drugs.
I work as part of the Neuropsychopharmacology Research Group at the Institute of Neuroscience in Trinity College Dublin. We're investigating the physiology underlying depressive disorder with a view to developing new and better treatments. Recent evidence suggests that another neurotransmitter system may be implicated in depression. MRI scans of depressed patients have shown altered levels of glutamate in some brain areas. Glutamate is the main excitatory neurotransmitter in the brain, but excessive levels of glutamate can be toxic to otherwise healthy neurons. In the healthy brain, glutamate levels are tightly controlled by cells called astrocytes, which take up and metabolize excess glutamate. Astrocytes also have other roles, including regulating blood flow within the brain. It has been suggested that altered levels of glutamate detected in the brains of depressed patients may be as a result of dysfunction in these astrocytes. This may lead to a damaging buildup of glutamate.
The aim of my research is to try and develop a series of magnetic resonance imaging markers associated with this astrocytic dysfunction. I performed scans on the brains of a rat strain known to exhibit astrocytic dysfunction to try and determine whether or not we can detect small changes in blood flow and brain structure.
If we can develop a robust set of imaging markers associated with astrocytic dysfunction in our animal models, we can use these markers to identify regions within the brains of depressed patients in which these cells are dysfunctional.
The development of neuroimaging markers associated with astrocytic dysfunction will give us a new insight into the underlying physiology of major depressive disorder. This should enable us to make new and more effective drugs and improve patient outcomes.